So in this presentation, I'm going to cover three topics. First one, I'm going to introduce the concept of myelin regeneration. Secondly, describe how this process changes with aging. And thirdly, talk about some experiments where we've been able to overcome the effects of aging, which of course has therapeutic implications. So one of the reasons we should wear a cycle helmet when we go cycling is that if we damage any other part of the body other than that protected by the helmet, the chances of regeneration and repair taking place are pretty good. By contrast, the structure that is within the cranium, the central nervous system, classically has a very poor inherent regenerative capacity. Now, while this is true of the neuronal component of the central nervous system, neurons aren't regenerated when they're lost, neither are their processes regenerated if they're severed. This concept of poor regeneration in the adult mammalian central nervous system is not true for another cell type present within the central nervous system, the oligodendrocyte. And this is the cell that makes the myelin sheaths within the central nervous system, which both allow rapid saltatory conduction and also maintain the integrity of axons over long distances remote from their cell bodies. So while there are diseases that primarily target the neuron, such as Alzheimer's and Parkinson's disease, there are also diseases where the primary target of the disease process is instead the oligodendrocyte. And top of the list of those diseases is the acquired autoimmune disease, multiple sclerosis. So when a maladaptive immune response occurs, as it does in MS, and kills oligodendrocytes, the oligodendrocytes are unable to support a myelin sheath which comes off the axon, and the axon is therefore demyelinated. Now, in contrast to the loss of neurons or any component of the neuron, the default response to loss of oligodendrocytes in the adult mammalian central nervous system, including the human central nervous system, is a spontaneous regenerative process which involves the generation of new oligodendrocytes that form new myelin sheaths, and both the architecture and function of the tissue is restored to normality. So this is a true regenerative response that is what one would expect to occur if you lose oligodendrocytes in a disease process. And indeed, this does happen in multiple sclerosis, at least in the early stages. Now, where do these new oligodendrocytes come from? Well, they come from a very abundant population of CNS progenitor cells, multipotent progenitor cells. One might even call them adult stem cells of the CNS. So this is a section through an adult mouse brain where the progenitor cells are labeled in both red and green. And you can see these are blood vessels, by the way. You can see within the adult mouse forebrain that these cells are extremely abundant. They're widely distributed throughout the whole of the central nervous system. And a similar picture would be true if one were able to obtain it from the human brain. They constitute about 6, maybe even up to 8% of all the cells in the central nervous system. So these are the cells that are responsible for generating new oligodendrocytes. And the process is very simple at one level. So in response to demyelination, the adult progenitor cell becomes activated, and that involves switching on a very specific bespoke set of genes that enable it to then undergo a process of recruitment, which involves their migration and proliferation, and then ultimately their differentiation into new oligodendrocytes. So that's how remyelination takes place. And in many ways, it's almost a paradigmatic example of spontaneous regeneration in an adult tissue. However, in multiple sclerosis, it's very clear that as one progresses through life with the disease, this regenerative process becomes less efficient. And instead, there's a tilting of the balance of outcome towards a failure of regeneration. And because one of the features of the myelin sheath is to maintain the integrity of the axon, the failure of myelin regeneration leads to irreversible loss of the neuronal element of the central nervous system, which is the anatomical process that underpins the eventual progressive phase of the disease. So one of the questions we and many others in the field have asked is, why does this regenerative process become less efficient with age? Why do you go from regeneration progressively towards irreversible degeneration? Now, it struck us a while back that in fact the answer to this question is at one level remarkably simple. So the single most important determinant of regenerative efficiency in any species and any tissue is the process of aging. And the same is absolutely true for regeneration of myelin. It occurs very efficiently in young adults, but becomes progressively less efficient as one progresses through adult life. So our hypothesis is that as a result of aging, some intrinsic changes take place in the adult progenitor cell, which render it less efficient at engaging in the various steps of the minor regenerative process. So in order to understand the failure of remyelination that occurs with aging, we really need to know something about how this progenitor cell changes as you progress through adult life. What seems to be clear is that the number of progenitor cells per se doesn't change. The number of progenitor cells you have in a young adult brain is much the same as the number you have in an aged adult brain. But clearly, their functional properties decline. In order to get a handle on that, it would be very useful if we could isolate these cells in tissue culture. And this has been, excuse me, this has been a technical block in the field for a number of years. So most of the studies that have been published on the progenitor cell involve isolating cells from neonatal rodent pups. There are a very small number of studies in progenitor cells obtained from adults adult in the sense that they're sexually mature, so 1 to 2 months of age, which in human terms is mid-teens, which is an age that we know is not associated with remyelination failure in clinical disease. So what we'd really like to do is to obtain these progenitor cells from really old rats, which would equate in human terms to the point where the disease is tipping from the relapsing, remitting regenerative phase to the secondary progressive phase where regeneration fails. Now, isolating progenitor cells from these aged rats was a technical challenge which was solved by Bjorn Neumann in the lab, who developed protocols, unique protocols for identifying, for isolating progenitor cells from any aged rodent that we chose to use, including animals as old as 22 months. And once we've isolated these cells, we can now look at how their properties change with aging. So the first thing we noted is that, as one might expect, the aging progenitor cell acquires all the hallmarks of aging stem cells. As, for example, increase in DNA damage, increase in senescence. Most striking, however, was the failure of these cells to differentiate into oligodendrocytes in tissue culture. So there are well-established protocols for inducing differentiation of progenitors into oligodendrocytes in a tissue culture dish. For example, the addition of thyroid hormone. So young adult progenitor cells will differentiate with some efficiency, and that could be pushed by thyroid hormone. But the aged progenitor cells find it extremely difficult to become oligodendrocytes, even in the presence of an inducer of differentiation, such as T3. So just by virtue of these cells being older, their ability to become oligodendrocytes is declining. Now, over the last few years, there have been a number of high-profile papers published using various screening strategies to identify small molecules that promote progenitor differentiation to oligodendrocytes. Here are 2 of those. One from Paul Tezar's lab, which identified this compound, miconazole, and one from Jonah Chan's lab, which identified benzatropine. And both of these compounds are being touted as potential regenerative medicines for promoting myelin regeneration, preventing axonal loss in progressive MS. So if you take young adult progenitor cells, these cells can be induced differentiation using these 2 compounds and a number of others that have been that are known to induce differentiation. Thyroid hormone, again, for example, and also agonists for the RXR receptor, which is the basis of a clinical trial that is taking place in Cambridge based on some work that we published a number of years ago. But if you take aged progenitor cells, these cells fail to respond to any of these inducers of differentiation. So here's a dilemma. As you get older and your need for regenerative medicine increases, the ability of your stem cells or progenitor cells to respond to those inducers of differentiation is declining. Now, is it possible to do anything about the effects of aging on the progenitor cell? Can you rejuvenate them? Well, we showed that as a point of principle a number of years ago using the parabiosis, heterochronic parabiosis model, where we showed that if you bathe the environment of an aged central nervous system with factors derived from the circulation of a young adult animal, the aged progenitors could be induced to regenerate as efficiently as young progenitor cells. So in principle, the effects of aging in the progenitor cell are not set in stone. They can be manipulated. They're plastic. So now we have the ability to isolate these cells from young and aged progenitor cells. We can do all sorts of omic studies and look at pathways and well, anything we choose actually that might change in the aging progenitor cell population. Here's just some examples of the changes that we see in the progenitor cells as they age. And one of the things that struck our that attracted our attention was changes in nutrient signaling pathways such as mTOR. Because it is well known that one of the most effective ways to reverse the effects of aging is to induce calorie restriction. So we did an experiment where we took aged animals, and over the next 6 months, we fed one group ad libitum, and the other group we calorie restricted by alternate day fasting. And then at 18 months of age, we induced demyelination and looked at the regenerative process. And whereas the regenerative process was as slow and inept as we expected to be in the controls, in the calorie restricted animals, the regeneration of new myelin sheaths was complete. These are non-overlapping groups. The difference between the ad libitum fed animals and the calorie restricted animals was black and white, night and day. So the calorie restriction, highly effective at rejuvenating failed remyelination in aged animals. Now that is not a very palatable or attractive way to reverse the effects clinically, and so we were challenged by our clinical colleagues to come up with a way that we could replicate this pharmacologically. And there are a number of so-called calorie restriction mimetics, one of which is metformin, the AMPK pathway agonist, which is now widely used in the treatment of type 2 diabetes. So in a wing and a prayer, we asked whether we could replicate the dramatic effects of calorie restriction using calorie restriction mimetic. And to our delight and surprise, we discovered that in fact, ad libitum fed animals treated with metformin completely phenocopied the effects of calorie restriction. That's shown here. And so metformin alone was able to dramatically change the effects of aging on the regenerative process. Furthermore, it also recalibrated the aged progenitor cells, so they now become responsive to all of these pro-differentiation factors. So this is the untreated aged animals in response to a number of pro-differentiation signals. But if you treat them with metformin beforehand, they now become, whoops, wrong way, they now become responsive to all of those factors. So the aged progenitor cell has been recalibrated by metformin so that it responds in a way that a young progenitor cell would do to these potential regenerative medicine small molecules. And we know that the metformin is working via the AMPK pathway because we can use both pharmacological and genetic approaches to disabling AMPK, and metformin no longer has a rejuvenative effect. So just for the last part of this talk, I want to move on to some work that we published last year. And this is a collaboration with my friend and colleague in the Stem Cell Institute, Kevin Shalou. It was work done by a PhD student we co-supervised, Mikey Siegel. So the question we asked is, why does the progenitor cell change with aging? Is it due to an intrinsic aging process, or is it reflecting the age of the environment in which it lives, its niche? So to address that question, we did a very simple experiment and argued that if you took aged progenitor cells and put them into the young central nervous system, if aging was an inherent process, they would behave like aged progenitor cells. But if the progenitor age reflected the aged environment, the aged progenitor cells in the developing CNS would behave like neonatal progenitors of development. And in brief, the answer was that aged progenitor cells in the young central nervous system now both proliferate and differentiate with the same efficiency as the neonatal progenitor cells. In other words, simply by putting an aged progenitor cell in a young environment, you can make it function as if it was a young progenitor cell. Speaking very much to the notion that aging progenitor cells is in fact extrinsically determined. So what is taking place in the central nervous system with aging that might change the properties of the progenitor cells? So we linked up with Christian Franze in PDN, and Christian, using atomic force microscopy, showed that the stiffness of the central nervous system increases quite substantially during aging. So the aged brain is considerably stiffer than the young adult brain and considerably stiffer than the neonatal brain. Now, this was a very simple experiment where Mikey took decellularized brains from young and aged animals and put young progenitors on the aged matrix and aged progenitors on the young matrix. And what he found was that the function of the progenitor cell reflected the age of the substrate rather than the age of origin. So aged progenitor cells on a young brain substrate behave like neonatal progenitor cells and vice versa. Now, what is the feature of the aging brain matrix that is determining the function of the progenitor cell? Is it ECM integrin signaling, or is it, does it actually relate the stiffness per se, the physical properties? So in order to address that, we used some chemically inert physical substrates, acrylamide gel substrates that Kevin had developed in his lab, and showed that actually the stiffness per se was able to change the properties of the progenitor cell. Age progenitor cells on a stiff substrate behave like age progenitor cells, but on a softer substrate mimicking the substrate stiffness of the young brain, they now behave as if they're young progenitor cells, both in terms of their proliferation and differentiation. And we went on to show that this was mediated by the mechanosensitive ion channel piezo1. And if you delete piezo1 from age progenitor cells, they are no longer able to sense that their environment is stiff and start to proliferate and differentiate as if they were young progenitor cells. So you disable the age progenitor cell from recognizing the stiffness of its environment, and it rejuvenates. And we did this both in vitro, which is shown in this slide, and also in vivo using a systemically delivered AAV to specifically delete piezo1 in the progenitor cell population. And we're able to show by doing that in the age brain, the differentiation of progenitor neurooligodendrocytes was increased. In other words, simply by removing piezo1 from the age progenitor cell, we were able to enhance remyelination. So this makes piezo1 and perhaps even small molecule inhibitors of piezo1 another credible target for promoting remyelination. So just to finish this presentation and throw a slight spanner in the works. So all of the work that I've described to you up until now, both from our own lab and other labs around the world, has really been focused on promoting differentiation progenitor cells in the age central nervous system as a way of driving the regenerative process in the progressive phase of MS. Now, in some very recent experiments by Bjorn Neumann and Sarah Forster in the lab, we've asked whether, in order for a progenitor cell during remyelination to differentiate, does it have to go through a round of proliferation? So is entering the cell cycle a prerequisite for differentiation? Or is it possible for a recruited progenitor cell to just differentiate directly into an oligodendrocyte? Well, it turns out that if you EDU label proliferating cells at the time of lesion induction, then you find that all of the new oligodendrocytes, which you identify with the marker CC1, are EDU positive. In other words, all the new oligodendrocytes have come from proliferating progenitor cells. Now, does that mean that they have to go through cell cycle in order to differentiate? Well, it seems that indeed it does. So if you do a tissue culture experiment and inhibit progression through cell cycle using lecodoxol, not only do you arrest cell cycle, but you also prevent these cells from differentiating into oligodendrocytes, even in the presence of thyroid hormone. And this has interesting implications, because if our progenitor cells in age CNS are in G0 and have been for some time, then a differentiation factor per se will not promote differentiation. Maybe that beforehand you have to kick those aging cells into cell cycle in order for them to go through the process of differentiation. So just to summarize, the efficiency of remyelination declines progressively with aging. And this is why there is a need for regenerative medicines in multiple sclerosis. And there's nothing heretical about that. The effects of age are profound in all regenerative processes. It is true that the aging progenitor cell does undergo intrinsic cell-autonomous changes, which prevent it going through the process of remyelination more efficiently. But those properties of the progenitor cell that are acquired with aging are reversible. And we've done this by a number of approaches, parabiosis, calorestriction, and also using small molecules. However, the effects of aging on the progenitor cell are, in fact, ultimately extrinsically determined. And we've shown in our recent work that this is in large part due to changes in the mechanical properties of the progenitor cell niche. And finally, it seems that actually, paradoxically, for a progenitor cell to differentiate, it has to have first gone through cell cycle and just recently exited the cell cycle in order for that to occur. So we may actually ultimately, in terms of therapy, need to be looking at ways not only of differentiating progenitor cells, but also initially inducing them to proliferate. So I should just thank the members of the lab who've done this work and the various funding bodies and various collaborators who've helped with us along the way: Bill Richardson, UCL, Amy Wages from Harvard, and Kevin Schlew from our own institute. And thank you very much for listening.